Good morning, and thank you for joining us for this webinar on cellular viability. My name is Nick Dolman. I'm a staff scientist at the Molecular Probes Division of Life Technologies here in Eugene, Oregon. Today I'm joined by Taryn Jackson from our technical support staff, and she'll be handling any questions you pose online throughout the seminar, so please feel free to pose those questions. This is the first of 4 seminars on cell health, and today we'll be talking about the basic changes that happens when a cell goes from living to dead, and ways to read that out. So when we say cell health or viability, we can mean a number of different things, but generally it's a change for the worse induced by one of the following. It can be a mechanical stress, a drug treatment, genetic alteration, for example, a knockout of a gene with siRNA, or indeed an environmental stress. And while sometimes that change towards a more of a dead phenotype is beneficial, especially in the scenario of cancer, we do normally look at trying to keep the cells alive. So I'll normally refer to death as a bad thing for a cell, but please keep in mind that it might not necessarily be bad in your particular setup. We'll look at cell health, but really at the highest level today, we're looking at whether or not a cell is live or dead. I'm sorry about this. My presentation is just frozen, so I can't actually move the next slide on. I'm just waiting to get the presentation back. Okay, there we go. We're now down to what we're going to be covering today. I apologize for that slight technical problem. So we're going to be talking a little bit about the parameters of cell viability and what are the ones most important to measure. And within that, we'll also talk about some of the properties that accompany cell viability. We'll then move on to look at some of the probes used to investigate cell viability. And we'll be concentrating on those that can be used in flow cytometry or fluorescence imaging. And we'll look at some probes that look at membrane integrity, ones that can read out some enzyme activity. And these are enzymes that are general indicators of cell health. We'll also touch briefly on how to combine some viability probes to get a more sophisticated and in-depth feel for the health of the cell, although these topics will be covered again in greater detail in our later seminars covering apoptosis, autophagy, and then pre-lethal indicators. And then as part of that sort of combining the viability probes, we'll look at some of the next steps, which can be adding something extra on top of a viability readout. And then finally, we'll finish up with a section from our technical support team, where we look at tips and tricks to optimize your labeling and detection, remove some of the background, and achieve the best results. I hope everyone can hear me okay. But if you can't, if you could just pose a question, I'll try and speak up a little bit. I'm not sure if we're having a bad audio connection as well as some of the problems we're having with the live meeting. So as we go down to cell viability properties, again we look at this sort of continuum of cell health as we go through from cells that are considered to be live to those that are considered to be dead. Now, you know, there are a number of markers that indicate cell viability. A live cell, one that's healthy, is proliferating but not hyperproliferative. They have intact membrane potential. They have enzyme activity. Of course, their plasma membrane is intact, or they have membrane integrity. When you're looking at cell health, you really want to look at something that's irreversible, because there are many different levels and very many different properties and parameters. But you really want to look at something that poses an irreversible change, and so that's why we tend to focus on membrane integrity when looking at whether or not a cell is live or dead. You know, there are a number of properties that can indicate death, and they're very specific to a particular cell type. And so if you're looking for one general overarching indicator of cell death, it's this irreversible loss of membrane integrity. So what are the parameters that can read out an irreversible change from something that's leading to death? Well, as I said before, we're really looking at whether the cell is alive or dead. Is it an irreversible change? And these can be read out in two main ways. One can look at plasma membrane integrity with reactive dyes and membrane impermeant nucleic acid dyes, and then we'll often add in a measure of enzyme activity. And most commonly, we're looking at the general cleavage of the AM ester groups of dyes such as calcine AM, and this gives a nice sort of 2-point marker of whether or not a cell is alive or dead. Okay, as we start to go into to probes that can be used to read these sections out, and there is a little bit of redundancy here. We're going to sort of stress over and over again that you're really looking at an irreversible change in plasma membrane integrity for live and dead, and then at the end we'll just reiterate that point with some examples of what you'd actually see with some of the assays. And so these viability detections, they are relatively simple. There are only a few ways of doing it, but that does make it nice that you can easily multiplex with other more sophisticated tools to maybe look at if your cell died via apoptosis or autophagy. Okay, so these probes, one of the ways that you can look at membrane integrity is by using reactive dyes. Here we've got a flow cytometry plot, and what we've got is heat-killed Jurkat cells mixed in with just regular untreated Jurkats. And using a reactive dye, you can probe whether or not they're live and dead very easily. And I'll just on the next slide go back to the theory of how you do this. So essentially, when a cell is live, the plasma membrane is intact, and the dye is only free to bind to proteins on the cell surface. However, if the plasma membrane is compromised, the dye can get in, and so they can react with all the cellular proteins. And this gives rise to a much brighter signal, which on that flow cytometry plot on the previous chart, you can see why the dead cells are, 2 to 3 log orders higher fluorescence than the dead cells. Now, it's important to note that this will not work in serum, so you will have to do these experiments in serum-free. They can work for imaging, although they are a little more difficult to use, so we really only recommend these tools as probes for use in flow cytometry. And indeed, because they're so useful for flow cytometry, and because flow cytometers can accommodate a wide range of fluorophores, we have quite a number available that really span the visible spectrum into the near infrared. So as we look at the second means to read out cell viability, and specifically membrane integrity, you can use cell impermeant nucleic acid dyes. And so again, the concept is the same as with the reactive dyes. If a cell is live, the plasma membrane is intact, the dye cannot get in. However, if a cell is dead, the plasma membrane's been compromised and the nucleic acid dye can get in. And so the advantage of this approach is DNA binding dyes are typically fluorogenic, and so you can perform these as no-wash assays. You can do them in the presence of serum. The reason why they're fluorogenic is when the nucleic acid dye binds to the DNA, it could be a minor groove binder, intercalator. Essentially, normally these dyes have free rotation around a carbon-carbon bond, which allows the energy to be lost without fluorescence. However, when they bind to DNA, they're locked into a rigid structure, which gives rise to a very bright fluorescence from the dye. And so these are particularly nice tools for using in a wide range of scenarios. Of course, one of the problems with any of these membrane integrity dyes is when you're picking up dead cells, you'll see a signal, but if you've got a completely live population of cells, you may not see anything, and so it's good to check for false negatives. And so we typically combine a membrane integrity dye, such as a nucleic acid dye, with another label. Both these examples here, we've stained cells with calcine AM, as well as with 2 different nucleic acid binding dyes. On the left we use Epithidium homodimer 1, and on the right Propidium iodide. So the green cells are living. Essentially what has happened is the calcine AM has got into the cell, and it's the active enzymes within the cell have cleaved off the AM groups, giving rise to a fluorescent product, and then they're well retained within the cells. The dead cells can't retain calcine AM, and nor can they cleave the dye, but they also show up very bright for these red nucleic acid binding dyes. And again, we have a range of these that span the spectrum and give you ample opportunity to multiplex them with whatever label you might have put in. Again, just down on the right we have a flow cytometry plot, and it's important to note that these do work for imaging and flow cytometry. And you can see that the dead cells that show up very bright on the right peak, and the live cells show up as a dimmer population to the left. And so I mentioned just briefly on slide 16 about the enzyme activity, so just to go into a little more detail here. Here we're probing enzyme activity, specifically using esterase substrates. These can get into the cells, and then the esterases cleave the AM group, so this is a common loading strategy we use to get dyes into cells. They become fluorescent. And again, live cells will be brightly fluorescent, and the dye will be retained, whereas a dead cell will not show any fluorescence because the dye cannot be retained within the cell. And again, like with many of our probes, we have a wide range of colours available. So here we've got from blue through to a reddy orange. And calcine green is one of the better ones of these dyes. It works really, really well. Some optimizations needed for both the blue, violet, and the red and orange as well, but they all can be used as quite effective labels for living cells. So now I just want to acknowledge some of the pitfalls, especially in the imaging of these probes, and actually talk about a new product that was released a couple of months back. So whilst you can use calcium AM and dyes like ethidium homodimer 1 or propidium iodide, their spectrums tend to be not particularly amenable to imaging. They have very broad emission peaks and not always optimal excitation properties either for all microscopes. And so we basically developed a new red dye for looking at membrane integrity, which is perfectly matched for Texas red filters. Of course, calcium AM, the green dye is perfectly matched for your FITC channel. That's a GFP or an Alexa 488 type of excitation emission properties. It works on the same principle that the red dye is excluded from live cells, however can get into dead cells. And then, of course, we use calcium AM to read out whether or not, to give you a feel for the live cells as well. Okay, and now we're going to talk a little bit about adding to viability. So there's a number of different mechanisms that you can add to look at an extra parameter over and above viability. And as I say, we'll cover these in a lot more detail in our forthcoming seminars on apoptosis, autophagy, and also pre-lethal indicators. However, I just want to highlight a couple of examples today, just to give you a little bit of a teaser, but also so you don't come away from this webinar with just knowing about live dead. One way that you can add to viability is actually to look at the nuclear condensation that occurs during apoptosis. And here we have an example using flow cytometry. On the left, we've got control cells. And indeed, if you look in that upper right quadrant, you can see that red population, and they are dead cells. So it's important to note that even in a healthy population of cells, you'll often see a few that are dead. However, the bulk of the cells are down in the bottom left and are showing very weak fluorescence. Here we're using Cytox A80 Advanced for dead cell discrimination, and that's a typical fluorescence emission that you'd see from vibrant dye cycle for living cells. If we look over at the right panel, we've got some camptothecin-treated gercats, and what you see is those green population down in the left are moving to the right into that blue population. However, they're still not dead. Their membrane hasn't been their membrane integrity hasn't been compromised, so you can still see that they have equivalent cytox staining as the live cells. But what you do see is an increase in the vibrant dye cycle fluorescence, and this is telling you that the nuclei, the chromatin, is being condensed, and that's a marker of apoptosis. Now, these can be discriminated against from your normal DNA segregation that you'd see throughout the cell cycle, and if one was to plot frequency against just raw vibrant dye cycle fluorescence, you'd see that G1S, G2M peaks being discriminated from these apoptotic ones. And in these camptothecin treated, you can see that apoptotic population marked in A and blue will then transition into that necrotic death, where the fluorescence of vibrant dye cycle violet increases, as does the cytox stain, showing that now the membrane has been compromised. Here's another assay that we commonly use in imaging to look at multiple parameters of cell death. On the left we have control, and these cells are labelled with the mito health stain, and that tells you the general health of the mitochondria. They're a good pre-lethal indicator of a cell's health. Polarized mitochondria indicate a healthy cell. However, if those mitochondria depolarize, it's a good indication that all is not well with the cell. So what you see in blue is Herc staining, and then in red you've got the mitohealth stain. And then in green you can see just a couple of dead cells. So again, this just highlights that even in the control population you'll often see a couple of dead cells. However, if the cells are treated with 60, should be micromolar valinomycin. Obviously, if you hit them with 60 millimolar valinomycin, you'll also kill them, but they were actually treated with 60 micromolar there. You can see a drastic loss of that red mitohealth stain, as well as the appearance of a lot more green positive cells that are showing that the plasma membrane is being compromised. And indeed, using on the left here, we've got dose response of this valinomycin-induced cell death, this time in HeLa cells. And you can see as you get more and more concentration, higher concentration, you get more and more death. There are other ways of reading out cell death as well, and with other parameters. So here, Cyquant Direct is an assay that tells you a little bit about proliferation state of the cell as well as the overall health of the cell. And it again can be used nicely in plate readers or high content images to look at dose response curves for various compounds and their potential toxicity to cells. Okay, now we're going to move to potentially what might be one of the most useful parts of the talk, is the cell viability tips and tricks. Okay, so it's important to develop viability controls because oftentimes if you're using an experimental compound or an experimental siRNA, you won't actually know how much death there could be, and so you won't know if your signal represents massive death or just a small change in one or two cells. So it's often important to have those positive controls for the death. So that's basically you're trying to have a condition that will kill the cells. Now, there are a number of ways of doing it. However, the ways that we recommend are heat killing, so you can place cells at 60 degrees for 20 minutes. You can also use alcohol killing. Actually, the advantage here, the cells can be stored for several years. What you do is you can centrifuge the cells and fix them. If you're using adherent cells, you can trypsinize them first. However, when you're trypsinizing them, just treat the cells as you usually passage them, pellet and remove the supernatant, and then add ice cold 70% ethanol, and that will kill the cells pretty nicely. And the added vortexing will ensure that they're dead. And you can resuspend them and store them. One of the nice ways when you have a population of completely dead cells is you can actually create a gradient by mixing live and dead. So if you wanted 50% live, 50% dead, just add equal volumes of your experimental sample to your viability control. Sometimes it's not particularly easy to do an alcohol kill or a heat kill if you're in a plate format or you've maybe got dishes. So you can also just use very high concentrations of a drug like camptothecin at about 40 micromolar for 24 hours, or 100 micromolar valinomycin for sort of 5 to 6 hours, and that should give you a pretty good complete killing of a population of cells. And as I say, that's often easier if you can't subject one part of the plate to heat, or if you're not able to use ethanol in a very controlled fashion. I say when it comes to using the non-permeant nucleic acid stains, it's important if you are ever trypsinizing or having to scrape the cells, this will disrupt the plasma membrane, and so that can give you a false positive on a dead cell indicator. So allow them time to recover, and for that membrane to just sort of relax and get back into its normal working order, and then you shouldn't get false positive readouts. When using dyes like the calcium AM, make sure there's no serum in the in the loading solution. So either use serum-free or a salt-based physiological solution, as the presence of serum will affect AM dye loading. And this is also true, as I said before, for the reactive dyes that can be used for flow cytometry. Again, they won't work in the presence of serum because the reactive dye will just basically bind to the serum and not your cells. Again, avoiding phenol red in the imaging solution. Whilst it doesn't have that much of an effect most of the time, it's always important to consider that it may be causing an effect if you're dealing with very low fluorescence or you're looking at very subtle changes. So it's possible to use to acquire phenol red free media, or use a DPBS or HBSS or Krebs-Ringer solution. Nucleic acid dyes, they do have an off rate, and so often that binding isn't, it's reversible, and so it will start to come off. So it's important to image the signal soon after you've done the labeling. And they're not formaldehyde fixable, so if sometimes fixation with formaldehyde will actually cross-link the dyes to the structures of interest. Now, this doesn't happen on nucleic acid dyes, so they won't be fixed in place. However, you can use them in combination with formaldehyde-fixed samples, so you can obviously use antibodies if you wanted to look at other indicators, things like cleaved caspase or LC3B staining for autophagy. So AM dyes, so your loading times and concentrations can vary very wildly. And oftentimes when you're looking at our product information sheets, we'll give you a range of concentrations and times to try, but remember often cells that are acutely isolated from animals do take a little bit longer, whereas cells growing in culture can take a little less time. And always just do your own empirical optimization of loading to ensure that you're getting the absolutely best signal. And when you are heat killing or using alcohol, you're pretty much killing those cells very quickly, so they're not undergoing apoptosis or they're not undergoing an autophagic cell death. They're pretty much undergoing just sort of necrosis via a mechanical insult. You're just basically permeabilizing the cells pretty quickly. Okay, so if we just take a little bit of time to recap on the expected results. So if you're using a plasma membrane integrity readout, you're looking at reactive dyes, then your live cells will be dim or negative, and your dead cells will be very bright and give you a positive staining. Same is true for nucleic acid dyes. Your live cells are dim and negative, whereas your dead cells are bright, positive. If you're looking at enzyme activity, then of course the flip is true. Your live cells will be bright and give you rise to a positive signal, whereas your dead cells will be dim and give you a negative signal. If you're using some of our dual color live dead kits, then the live cells are green and the dead cells are red. Okay, so just to finish up for today, just to remind you of our technical support. If you are using these assays and do need any help, we have teams available to speak online or on the phone to help you with your problems. And also we do have upcoming seminars that will go into more details on some of the mechanisms of cell death, looking at probes we have for apoptosis and for autophagy. And then we'll also have a fourth seminar which will look at some pre-lethal indicators, where we'll look in more depth at things like generation of reactive oxygen species, mitochondrial health, as well as various other parameters. So I'd like to finish off today. We'll be online for another 5 or 10 minutes just to answer any questions that come in if you didn't want to type them as you were listening. We'll be online, and please feel free to pose those. We will be silent once the webinar finishes, but we will still be online to answer the questions. And we could also get back to you via email as well if you need a more detailed response. So I'd like to finish up and thank you all for attending, and hopefully we'll see you all at some of the upcoming seminars in the future. Thank you very much. Goodbye.